we're fortunate that we have folks like Syama Chatterton in the civil service paying attention to these things. But root diseases and soil-borne pathogens are something that have really reared their head in the last couple of decades and are seriously threatening pulse production in particular. Not so much soybeans, but they're not free and clear either. And so Syama is here to talk about a complex tale, root rot of pulses. Dr. Syama Chatterton joined Agriculture and Agri-Food Canada at the Lethbridge Research Centre in July of 2011. And she's a pulse crop pathologist. Her research focuses on management of root and foliar diseases of pulse crops and molecular diagnostics and identification of soil-borne pathogens. Dr. Chatterton is chasing the dream of developing a robust decision support system to reduce root rot risks for sustainable pulse production and those producers who love their soil. Syama.
Okay, hello everyone and good morning. I'm really happy to be here from Alberta. Caught the flight out yesterday. It was good timing because I think the flights from out of Alberta had been canceled for 4 days in a row previous, so I got here just in time. And then hopefully I can get home because there is a snowfall warning for Calgary and Lethbridge area today as well, so we shall see. Yeah, so I love pulse crops. I guess that's why we're here, why Manitoba Pulse Growers is supporting the session, because we all love pulse crops. I kind of like them because they have really pretty flowers. They're not like wheat, not like cereals. You don't even get to see the flowers. So they're really pretty. You get to get out there and see them. And the reason why we really love pulse crops is because of what we've been talking about this morning and that Yvonne really nicely introduced was their ability to fix nitrogen. Here's a healthy root system. I don't get to deal with these very much as a plant pathologist, but we can see just these massive amounts of nodules that can be present on these pulse crops. And this is why we want to put them in our crop rotations, why they're an integral part of climate change solutions, and why I think the pulse industry wants to see that increase of arable acres cropped to about 25% of pulse crops, which would mean a pulse crop in your rotation once every 4 years. There is also an increasing demand worldwide for pulse protein. Whether or not you sort of agree with replacing meat protein with pulse protein, you can see that there's really a global push towards that. And in the report on climate change solutions, it was recognized that consumption of healthy and sustainable diets presents some major opportunities for reducing greenhouse gas emissions, and a lot of that comes from pulse crops. And because of that, we know that soil health and pulse crops are very closely linked. So what is a healthy soil? I thought it was very interesting that Yvonne presented the very same definition, and that kind of saves me a little bit because I have a lot to talk about, so now I don't have to get into what a healthy soil is. From my perspective, when we're out in the fields digging up thousands of roots every year, I think a healthy soil is if you can get the shovel into your ground without having to jump on it about 10 times to dig up your hard clump of soil. But there's many different soil health parameters. Again, Yvonne talked about them. And I think what's interesting is when you look at this whole range of soil health parameters, what we often don't see is just a measurement of plant health. Like, how is your plant actually growing in that soil? So we try to collect a lot of data from the soil to tell us how our plant should be growing. Sometimes we just need to go and look at how that plant is growing. And so I guess the question is, if by all your soil health parameters, you're being told that you have a healthy soil, can you have soil-borne pathogens in those healthy soils? Can you have diseased plants in those healthy soils? So by definition, that soil is no longer healthy because it is no longer sustaining plant growth. So I sort of thought of this as like an oxymoron and be like, whoa, deafening silence in the room as I drop the mic on this. Well, you can't have a healthy soil if your plants aren't healthy. But most soil health parameters are not accounting for soil-borne pathogens. It's not something, and maybe Matthew will talk about this in the panel, But when you look at all those lists of soil health parameters and things that you can measure, you never see presence of soil-borne pathogens in that list. Partly, it's very, very difficult to link what some of these soil health properties are in terms of the chemical and the physical structure with presence of soil-borne pathogens. And so we're left with kind of this conundrum with pulse crops, where we know that pulses, I don't know if that works, not really. So we know that pulses contribute to healthy soils. But on the flip side, we also know that pulses tend to get a lot of soil-borne pathogens. So somewhere in there, there's this disconnect between what's our healthy soil and why is it supporting soil-borne pathogens. So I don't have the answer to that. I wish I did. I could stand up here and give you exactly why do healthy soils, why do they get soil-borne pathogens? I don't know. I'll get to it right at the very end of my talk, maybe with some of the things that are going on. But what I'm going to mostly talk about then today is what are those soil-borne pathogens that affect pulses, and what are some of the soil properties that are affecting them. So when we talk about pulses, for Alberta, Saskatchewan perspective, we have peas, we have lentils, we have dry beans, we have faba beans, and then we also have soybeans. Is it a pulse crop? Is it an oil seed? For Manitoba pulse and soybean growers, they're all lumped in together. And so those are kind of the 5 pulse crops that we will talk about today. But I'm really a pea. I love peas. That's really my area of specialty. So I'm going to focus mostly on pea root rot. I grew up in Vancouver. One of the favorite shows in the 90s was Da Vinci's Inquest. I don't know if anyone ever watched that. It's about this really roughshod sort of coroner that was trying to get to the truth of all the underseed of Vancouver's criminal scene. So instead, I've kind of changed it to my Syama's Inquest, where we're talking about in the underground of pulses, every pathogen has a secret. So we're trying to dig into what are the secrets of these pathogens, and how can we use that to then manage pathogens. So what I'm going to talk about in my inquest over the next 30 to 40 minutes is first looking at what are the pathogens that cause pea root rot, and how do they interact with each other. What are some of the other pathogens on pulse crops, and do we get pathogen crossover between those different pulse crops? How can we link root rot and soil health? And then finally, probably what everyone is here for, but it'll be the last 5 minutes of my talk, is what are some of our management options. So we'll get right into pea and lentil root rot. And this is kind of lump in lentils with peas because for the most part, they have the exact same problems with root rot, the same pathogen complexes. And what we find when we go out into the field looking for root rots is that I'm going to show you pictures later on of these amazing symptoms of Aphanomyces and amazing symptoms of Fusarium. But when we get out into the field and we start looking at what's going on, you really can't tell. The roots end up looking, don't know if the laser pointer, your roots often look like this or like this, and there's just kind of a mess of different pathogens going on. And determining kind of the symptoms, or determining what pathogen you have based on the symptoms can be very difficult. And so we have a number of players in our underground crime scene here. And these are the root robbers. And starring our number 1 is Aphanomyces euteiches. And I will tell you about that in the next slide. We have the henchmen, Fusarium avinaceum and Fusarium solani. A little bit kind of OCD sometimes about my presentations, and I spent a lot of time making sure I had exactly the right kind of animations to represent kind of the characteristics of these pathogens. And what we find is that Aphanomyces is kind of our A-list player. It just gets in there by brute force and punches its way into the roots. Fusarium avinaceum, I kind of think it's a little bit like, it doesn't quite know what's going on. It's there because it kind of hitches a ride in, but it can cause a lot of problems. Fusarium solani is a very targeted pathogen towards peas, so I kind of have it like that nefarious look. And then some of our other ones, Pythium, other Fusarium species, Rhizoctonia, we just kind of find them, they're like the hapless guys that fell into crime by accident. They don't really know why they're there. They're just kind of hitching a ride along. And then what we find is it's produced by wet soils, pulse history, and very tight rotations of pulse crops. So our big bad in this scenario, like I said, is Aphanomyces euteiches. This really exploded onto the prairie crop scene about 10 years ago. It wasn't really on our radar as something to watch for. We thought everywhere else in the world had a problem, but Canada didn't, partly because we thought that our soils were maybe too cold in the winter for this pathogen to overwinter. But what happens is that as you grow more and more pulse crops, it increases its presence in the soil more and more until you reach that threshold. And so we kind of reached that threshold in a lot of places in 2013, where all of a sudden there was a lot of fields, particularly in Alberta and Saskatchewan, that looked like this one here. 2013 was a very, very wet year, and we just saw a complete collapse of some of the pea fields. And then when you pull up the pea plants, you can see right away they're not healthy. You got very yellowing pea plants. You really don't have any root systems left at all. And then when we look at what Saphanomyces doing in the soil, the biggest problem with this pathogen is that it produces these oospores. They're very thick-walled resting spores. You can see them here in a root, and you can see that they have this really thick double cell wall with this really fat kind of lipid granule in the middle. So you see sort of the dense area in the middle that just lets them sit in the soil and survive until you get rain, or you get pea crop plus rain. So that's the recipe for these oospores to kind of wake up, and they germinate to produce these zoospores, which are they're very short-lived, but they can swim. And so you have those little soil pores and little pores in your soil that can fill with water, and they will swim through those spores or pores to find the roots. And then you can see here's a big mass of them. So you move from having one oospore to thousands of these little zoospores that then swim around to find the roots. And then once they find the roots, they can infect very, very quickly, and you can get kind of complete collapse of your roots in about 7 to 10 days after infection. So what this means is that usually when we get our big heavy rainfalls in kind of mid-June, you can have, I think talking yesterday with some scientists, and we're saying, well, what we often see now is that all their rain events kind of come at once. Rather than have rain kind of, scattered across the growing season, you can sometimes get like 50 millimeters of rainfall right in the middle of June. And so that's kind of the perfect time to have all that rain, wake up these zoospores, and then you can see kind of 2 weeks later, you can see your pea plants start to go down. And so once they've used up, they've gone through all that nice juicy root material, they've used it all up, they go back to this resting stage because there's nothing left for them to eat, and then they just sit there and wait in the soil. But what's interesting is when we looked at kind of the distribution of these root rot pathogens over a series of 5 years, what we found is that Aphanomyces eutyches almost always co-occurs with other pathogens. You never just find it on your own. So here if we look at pea fields, 55% of them had Aphanomyces, and usually a Fusarium species. And then the other 45% just had Fusarium root rot. And so we saw the same thing with lentil, although a few lower, less fields were infected with Aphanomyces. And so what our other pathogens tend to be is the hench mean. It's a spelling mistake that I noticed. I tried to fix it, then there was a problem with my presentation, so I got the old version put up there. Anyway, I think it kind of works. Call them hench mean because they are kind of mean. They infect those roots. Usually what we see when there's only Fusarium present, you see that they really attack at the point of seed attachment, and you'll see this blackening of the taproot that then usually does spread. And as that taproot blackens, the lateral roots can't stay attached, and so you'll kind of see this loss of lateral roots overall. Oftentimes, if you do cut open the roots, you will see this reddening of the kind of vascular system. So that's telling us that these Fusarium species can actually get inside the root, they get into that vascular system, and then they also prevent water movement up through those channels. The disease cycle actually isn't quite as well known as Aphanomyces, which is interesting because Fusarium has been around for a long time. But what we generally find is, again, they start with these spores that survive for the most part on crop residue. So you get this sort of question of how is tillage affecting, or no-till affecting Fusarium, because it generally survives on residue. And then as the plants kind of grow through that residue, it's when they start getting infected. But the part that's unknown is that we do often find fusarium species up in the foliar parts of the pea plant. So you can find them on pea seeds, you can find them on the pods. So we're not really sure how they're getting from the roots up into the foliar part, and then how much that contribution of fusarium on your seed is then impacting fusarium root rot later on. So there is some work to do on kind of deciphering this aspect of the disease cycle. So what I'm really interested in then is that we have this sort of Aphanomyces root rot and we have fusarium root rot, and we often find them together. So how are these pathogens interacting with each other? And we can kind of see right away just in this picture of symptomology. You have Aphanomyces on its own that creates these honey-bound brown discoloration. We have a fusarium species here, Fusarium redolens, which is everywhere in the soil, but it's actually very weakly pathogenic. We can see we really didn't get much disease there. Then you put Aphanomyces and Fusarium redolens together, and I'll show you what happens in the next slide. But just looking at symptomology, it looks very similar to just Aphanomyces alone. And then like I showed you pictures of the Fusarium solani, where you can see that blackening of the taproot. And then you put them together, and then you start seeing the symptoms that we're seeing more in the field, where you get this sort of black discoloration, and then that honey brown discoloration from Aphanomyces. So what we found when we do co-inoculation studies is that disease severity increases with multiple pathogens, and I'll try to walk you through this with animation if it works. So what we see is that when we have Aphanomyces alone, it's quite a serious aggressive pathogen on its own, causing very high levels of disease severity if we're looking at 5 being the highest amount. Like I said, Fusarium redolens on its own really doesn't cause very much disease, but when you mix it with Aphanomyces, you actually end up getting higher disease severity than Aphanomyces on its own. So it's kind of interacting with Aphanomyces to make it worse. You see the same thing with Fusarium solani. On its own, it's it can cause a lot of disease as well, but you mix it with Aphanomyces and then disease is higher. Avanasium, we didn't see that effect, and that's because we were almost at the max level that you could see with Avanasium anyway, so we didn't get that sort of synergistic effect. And then when you look at all of the pathogens together here, just in comparison to if you only had Fusarium, again, you can see that that presence of Aphanomyces is making the disease worse, and that all those kind of 4 pathogens working together is really what's driving disease severity. And so we looked at how these pathogens are then colonizing those roots. What's interesting is that we can see that Aphanomyces doesn't need any help to colonize the roots. So when it's present with redolens, solani, or Avanasium, the colonization levels stayed the same. They stayed pretty high. With redolens, which was a very, very weak pathogen, you can see that its presence increased when there was any other pathogen with it. So the ones that are more effective pathogens, like Aphanomyces, are opening up avenues of colonization for redolens. We saw the same thing with solani, which was surprising because it is a fairly serious pathogen on its own, but again, it's being helped along then by this presence of these other species. Avanasium was interesting because it was the one where we actually saw a decrease in colonization levels when Avanasium was present with other Fusarium species. And we've been looking at this in more detail, and it has a lot to do with the toxins that are produced by Fusarium Avanasium and how it competes with other Fusarium species in particular. And then the other thing is that the pathogen composition does change over time. And what we find is that in the field, Aphanomyces hits pretty early. Usually we can find it kind of at its peak on the roots in June. And mid-June is when we go and sample. Usually try to target about 6 to 7 weeks after planting, and especially if we've had some rainfall in June. But then if you go back and check those roots around flowering, so about 2 to 3 weeks later, we can see those Aphanomyces levels really drop off, and that we really start to pick up Fusarium Avanasium. And this is what those change in symptoms look like. So this is what it looks like in mid-June, when you can kind of see that very characteristic Aphanomyces look. And then in mid-July, you can see that you really don't have a lot of root material left at all. So one of the keys is that if you're going to sample for Aphanomyces, you have to sample early. I think what happened is that historically we would be doing root rot surveys in July, because that's actually when you start to see the plants start to not look very healthy. but we were missing Aphanomyces because it had already reached its peak in the roots, and then these other pathogens were coming in. So if you're concerned about root rot pathogens in your pulse crops, you have to sample before you see the plants actually start to show some of those shoot health issues, which can be tricky because you don't want to just go out and collect roots. It's a lot of work collecting roots, so you don't want to go and collect roots when your plants are looking healthy. Usually wait till they look diseased to go collect roots. And so then I'm going to talk about next is how soil moisture influences root rot pathogens. And really what we've seen when we're looking at kind of the surveys that we performed in Alberta, Saskatchewan, and Manitoba is that the prevalence and incidence of Aphanomyces really goes up and down with soil moisture. I'm showing the data from Alberta just because we have a longer data set, whereas Manitoba, I don't have the kind of the up-to-date data. And so what we found is that in the wet years, which is 2014, 2016, and 2020, we had a really high prevalence of Aphanomyces. 2015 is an anomaly because in that year they were not random surveys. We only went and surveyed fields where producers called us and said, I have a problem with root rot, and then it happened to be Aphanomyces. But you can see that in 2015, that was a fairly dry year, our incidence. So that's the number of samples that were positive in the field were actually quite low. And so then we see this decrease again in 2017, 2018, and 2019. So, I mean, this makes sense because we know that Aphanomyces is really, really dependent on soil moisture. And so what we wanted to do is, okay, we know this sort of pattern of how Aphanomyces is presenting based on precipitation levels, but how is it interacting with other pathogens and these soil moisture levels? Kind of knowing that Fusarium species tend to like drier environments, whereas Aphanomyces tends to like the wetter environments. So what we did was a greenhouse study where we tried to have different soil moisture levels. It's not that easy to kind of make sure that your treatments don't overlap a little bit, but what we tried to do was have prescribed watering to reach a 30, 40, and more than 50% high volumetric water content. And the more than 50% was basically, the soils were saturated at all times. And then these were inoculated with Aphanomyces and Fusarium avinaceum alone or in combination. And then we looked at disease severity, root mass, and then pathogen colonization. And so what we find was that the soil moisture affects pathogens differently, and that in fact it infects Aphanomyces a lot more than Fusarium. And so what we can see is that we really got almost no disease severity with Aphanomyces under those dry conditions, but under that high moisture, we had very, very high disease severity levels. Interesting for Fusarium avinaceum, it didn't really seem to matter whether it was low, medium, or high, which is not what we were expecting because the literature says Fusarium avinaceum prefers kind of drier conditions. So it just shows that, under all these soil moisture conditions, Fusarium root rot was still a problem. But what was the most interesting and kind of the most depressing is that when we look at that combination of Aphanomyces and Fusarium avinaceum, we can see disease severity increases under all of these moisture conditions. So even though Aphanomyces doesn't like dry conditions, you mix it with Fusarium avinaceum and you see that the disease severity goes up. But it was the highest under that high moisture condition. We look at the effect on root weight, which is sometimes it's a bit of a better quantitative measurement. What we can see is a very similar pattern. These are our controls here. What was interesting, of course, is the effect of drought. So drought, we had less root mass to begin with. And then when we had Aphanomyces here, there wasn't really a big impact on that root weight, even though we saw a big effect of disease severity, with a slight reduction in the root weight under the high moisture conditions. With Fusarium avinaceum, we can see that the root weight was actually the lowest in the low moisture condition. So again, then that's saying, okay, well, there is some benefit to Fusarium avinaceum under those dry conditions. And then we can see it increases as the soil moisture went up. But we saw the most drastic reduction in root rate when both Aphanomyces and Fusarium were present under all three of the moisture conditions. And this is just what it looks like. So this is when we have Aphanomyces alone, Fusarium on its own, and then when you have all three of them together. And this would be from the high moisture condition where we can see we have the worst root rot. Okay, and then we look at what are those levels on the roots, and what this is telling us, measuring the levels on the roots, is telling us well, how much is being left behind then in the soil? How is it colonizing? And what does that mean for what's being left in the soil? And what we can see is that Aphanomyces really only colonizes under those high moisture conditions. So that's why moisture is such an important factor in root rot risk, because you're only getting significant amounts of inoculum left behind in the soil when you have high moisture conditions. And then what was interesting is that even though we saw that disease severity was a lot higher when we had both Aphanomyces and Fusarium present, we can see that Aphanomyces colonization wasn't actually a lot higher. Whereas if you go straight down and we can look at the Fusarium avinaceum here, we can see that its colonization was higher under low and medium. So it's the colonization of Fusarium that's driving that higher disease severity under those low moisture conditions. I'm kind of losing my voice, hope I could make it through. Okay, so what we're kind of looking at with the environmental implications of a pathogen complex. Yeah, so I sometimes feel like, in Alberta, they call me the Grim Reaper. I feel like I stand up here at a lot of talks and have to give kind of the bad news, because the problem with a soil-borne pathogen complex is that the environment doesn't matter as much. So root rot was worse when both pathogens were present, and it made those dry conditions that usually aren't that great for Aphanomyces. We can see that it still helps drive root rot because of that interaction with Fusarium avinaceum. So multiple pathogens increase the risk under dry conditions. But Aphanomyces biomass was only high in the roots from saturated soils, and that's when we expect to see that massive increase in inoculum to soil. And then Fusarium inoculum, on the other hand, increases under dry conditions. So you're kind of hooped either way. You get Aphanomyces in your wet conditions, and then you get Fusarium under dry conditions. We did some studies with temperature, and I'm not going to show the results because really it turns out temperature's not as important as soil moisture conditions. And really our soil temperatures, once they kind of hit that 15, 18 degrees, you don't get a lot of change over the summer anyway. So I'm going to switch over now to talk about what some of these other pulse crop options are. And because, like we've said, pulses and legumes are essential to building sustainable systems. So what's going on with some of the other pulse crops? And I have dry bean in here, but I think there's some research I just saw out of Manitoba pulse and soybean growers, right, that can kind of consider dry beans don't really fix nitrogen. So you have to fertilize them, but then there's some maybe recent research to say that they are fixing more nitrogen than was previously thought. So we're putting them in here as an important part of our soil health systems. And then of course we have soybean, and then a pulse crop that maybe isn't grown as much, but can have some advantages is faba beans. So if you have Aphanomyces in your soil, that's the first question I usually get is, what can I grow now that you know you have Aphanomyces? And for sure, you cannot grow pea and lentils for a number of years. I'll get to that in the management section. Soybean, faba bean, and chickpea are fortunately tolerant or resistant to Aphanomyces, so you can grow those, but I know that there's some questions about their adaptability. Soybean does well in Manitoba, but we don't see it do that well in Saskatchewan and Alberta. Faba bean might actually be a really good fit for Manitoba because it requires moisture, but it's not a great fit for Alberta. And then chickpea is really only a great fit for southwestern Saskatchewan. Dry bean, you can grow you can grow it if you have Aphanomyces in your soil, but it's actually susceptible to a different race of Aphanomyces. And I don't want to talk about that too much, but if you have Aphanomyces in your soil that can infect pea, it can't infect bean, so you can still kind of rotate pea and bean. Alfalfa, yes and no. Alfalfa also has races, different races of Aphanomyces. Some of the peas, it's a bit confusing. In the States, they do label their alfalfa varieties with resistance to race 1 or race 2 of Aphanomyces, but we don't in Canada. In fact, our Canadian cultivars aren't even tested for Aphanomyces. So there is maybe a gap there that we need to start looking at that, because I've had a lot of producers that say to me, well, I've never had peas, but I've come out of alfalfa and now I have an Aphanomyces root rot problem in peas. And it's like, well, it's because of this the alfalfa that you grew would have been a variety that was susceptible to the pea isolate. With interest in cover crops, I see a lot of people trying to include vetches in their cover crops. That's an absolute no. Vetches are more susceptible to Aphanomyces than peas. So don't use vetches in your cover crops if you are worried, or if you want to rotate with peas. Clover, it really depends on the species. Lupin, I think, is a new pulse crop that has a lot of interest. We've tested that in our Aphanomyces-infested soils, and they don't get infected, so that's also an option. When we look at fusarium species, it's not quite as clear-cut as the Aphanomyces, and that's because, for the most part, fusarium, both Avanasium and Solani, can infect most pulse crops, but they can infect it to different degrees. And so we tested them against soybean. You can see it really didn't get a lot of disease. But when we tested it against faba bean, it actually could cause a fairly high amount of disease. Chickpea, it was sort of moderate amount of disease, except for one cultivar, Leader, didn't even grow in the presence of Avanasium. It just completely destroyed the seed. And then lentil actually isn't as susceptible to Avanasium as pea is. Then if we look at, and the problem with Avanasium is that it's associated with Fusarium head blight of cereals and also canola root rot. It's not the major driver of Fusarium head blight of cereals, that's Fusarium graminearum, but Fusarium Avanasium, like I said, it just kind of like tags along, and like, don't know what it's doing and why is it everywhere. So there's just some, we're looking at kind of more into this isolate aggressiveness based on what crop species it came from. Fusarium solani, very similar story to Avanasium. It kind of has differential aggressiveness on different pulse crops, but in this case, again, caused disease on faba bean, not very much on soybean, but then it was a different chickpea cultivar, Orion, that was more susceptible to Solani, whereas Leader was more susceptible to Avanasium. And then again, lentils aren't as susceptible to the Fusarium species as pulse crops. And in terms of Solani, there is actually some very specific host specialization within pulse crops, so it could just be that we didn't test enough isolates, because you can have some isolates that can only infect lentil, some isolates that can only infect pea. The major root rot pathogens of soybean, this is from Young-Min Kim at the Brandon Research Center. A lot of different Fusarium species, and I think this is just showing kind of the variation of Fusariums that you can get from kind of one field of soybean. These are all the plates that they pulled out. So again, it's a lot of these different Fusarium species that are at play with our pulse crops. And then of course the other major one on soybean is Phytophthora root rot. I'm really not going to talk about it that much except for this slide. And then what's interesting is that Phytophthora is in the same oomycete group as Aphanomyces. So they're both oomycetes, they both produce those oospores, they're dependent on water. But whereas Aphanomyces likes peas, Phytophthora likes soybean. So that's sort of good news because it means we're not getting crossover of those two pathogens. And then the other good news for soybean is that they are resistant cultivars against Phytophthora root rot, but new races can develop fairly rapidly. So there's always a lot of research going on to make sure that we're on top of what races are present in the soil. Okay, similar to what we see for soybean and peas, dry beans, their major root rot pathogens are also these Fusarium species. So I think we're starting to sort of get the trend here is that Fusarium species just really tend to love pulse crops. And so a lot of the same Fusarium species that came up on the list of soybeans and that come up on the list of peas are also pathogens of dry bean. Faba bean, I think this is why it holds a lot of promise. So far, root rot has not been a problem. We have gone out trying to look for it, and most faba beans are fairly healthy. It could just be because we haven't grown them for as long. They're fairly new. When we do inoculations in the greenhouse, they are susceptible to those Fusarium species. But when we've grown faba beans side by side with peas to look at root rot, we really didn't see any root rot develop. Here's a picture here. We can see the peas have a lot of root rot, and those faba beans have nice, healthy, very juicy root system. And in fact, they provided a yield benefit. If you grew them before a pea crop, they actually provided a benefit to that pea crop. So we are trying to look into that a little bit more as to what that interaction is. So let's talk a little bit about that crossover of pathogens. And what we did was grow soybean and pea side by side at sites in Moreton and Redvers, and then tried to look at what were the shared pathogens between the two. First, we can see that at Moreton, soybeans yielded a lot better than peas in our two years here, which happen to actually be quite dry years. And then we look at the pathogen composition on soybean compared to peas. You can see peas had a lot of Aphanomyces in 2019 and also Fusarium avinaceum, whereas even though soybeans are susceptible to those Fusarium species, we really didn't see that same colonization levels. So again, encouraging. In 2021, the peas were sampled late, so we actually didn't pick up any Aphanomyces on the peas, but I can tell you that it was there. And in 2021, we actually found there was more Fusarium solani on the peas than Avinaceum, but again, soybean was not colonized by those same pathogens. And then when we look at red vers, I don't know what happened with the soybean yield here, but really good soybean yield in 2018, and actually fairly good pea yield. Then in 2020, I think, was an extremely wet year in red vers. The soybeans didn't yield at all. And red vers is maybe, it's in Saskatchewan, so we're getting onto that sort of limit of where soybeans like to grow. But we did get pea yield, and then in 2022, which was even wetter than 2020 in red vers, we actually did see some soybean yield, whereas peas yielded nothing. And then again, the similar story with the pathogens. We can see really, really high colonization levels of Aphanomyces in 2018 and 2020, and we didn't pick up any Aphanomyces at all on the soybean. And the same with those Fusarium species. They're there, but they weren't colonizing soybean. So I think that's encouraging that we're kind of recommending that if you have a problem with Aphanomyces in your pea crop, then you can safely plant soybeans, you can safely plant faba beans, and as well chickpeas. So, how much time I have left? Okay. Yeah. So we'll talk a little bit about root rot and soil health. There's not that much to say, mostly because we just don't know a lot about it yet. You know, we talked about this continuum of pulses producing healthy soils, but then I just showed you that soil-borne pathogens love pulses. So we've been trying to do some work, and not just me, but other groups as well, on trying to find what are those indicators in this soil that would say, okay, well, the soils aren't healthy because of these chemical and physical properties, so they're supporting soil-borne pathogens. And we just have not been able to find any kind of clear indicators in any of these soil health parameters when we look at kind of healthy and diseased soils from the same field, or if we compare healthy and kind of diseased soils from different fields. What's interesting is that pea root rot is equally distributed across soil zones. So we find the same level in the black, the dark brown, and the brown soil zones. But when we kind of look at soil type and look at some correlations with disease sites, we do find that there is a positive correlation with clay. So heavy clay in your soil will mean you're more likely to have a problem with root rot, whereas those sandier soils tend to have a negative correlation. The soil zone surprised us because that's really dependent on moisture, right? And I just showed you how important moisture was, and then it's like, well, when you actually look at how the pathogen's distributed across the prairies, it's present in all the soil zones. And then there's other correlations are kind of all over the place. We had one study where potassium levels were negatively correlated with Aphanomyces root rot, and then another study where they really had no effect at all. Same with calcium, that's been really heavily implicated in Aphanomyces root rot out of a study from Sweden. We looked at some of the same properties here in soils from Canada, and we really found that there was no effect. I think in general, we just have higher calcium levels in our soil, so it doesn't turn out to be as important. What was always positively correlated, though, was nutrients, which is interesting. But I think what's happening is that the diseased roots plants aren't being able to use the nutrients, so you kind of get like nutrients left behind in your soils. And then magnesium in one study was also negatively correlated, but we haven't really found many other things. We did try to measure compaction, but the soils were so compacted we couldn't even get the compactometer thing into the soil. But it was kind of the same in all the soils that we looked at because it was a really dry year, so all the soils were kind of, you just could not get that penetrometer through them. And then the other thing is kind of, sort of root rot and soil health and biotic properties. And I would say like the holy grail for all soil-borne plant pathologists is trying to find these disease-suppressive soils. There's amazing kind of records in the literature of when someone's found one. Take olive weed as a really good example. And so that's something that we're always chasing. And I'll show you in the next slide, maybe something exciting. But when we look at sort of the microbial communities that are associated with these diseased and healthy sites, again, there's been nothing really clear that stood out. except for pathogenic fungal groups, particularly the Fusarium species, do increase as you increase pulse frequency. But we don't know, like, why is that happening? Why do Fusarium species love pulses? I don't know. In terms of disease-suppressive soils, what we're looking for here is soils that no matter how much inoculum you add, they don't get disease. And we did find one, which is very exciting. You just have to figure out why it's suppressive. So this is just an example of showing kind of our typical disease response curve. If you add oospores, the higher you add oospores, the more disease severity that you get. Then we found this one soil that happened to be like 2 kilometers from the Lethbridge Research Center, that no matter how many oospores we added here, you can see this is non-autoclaved, we just did not get any disease. When you autoclave the soil, so to remove any biological properties, we can see that we still had fairly low disease, but then it did go up in those highest amounts. So that would tell us that it is biological in nature. And we do have some research going on right now trying to figure out if we can really see, like, what's the difference, what's the biology that's going on in the soil that's different from the other soils. So that's all I'm going to talk about root rot and soil health, because like I said, we maybe don't know that much about it yet. Maybe Matthew is doing more work on that he can talk about. So I'm going to talk with my last 3 to 5 minutes on management of pulses. Do I have time? Yeah, okay. Okay, so I hate this slide. It's my most depressing slide. So what are our management options for pulses, root rot of pulse crops? You know, if we're talking peas and lentils, we don't have genetic resistance. There's work on it for peas. There's some hope coming soon. Fungicides don't work because you can't spray something. You can't spray foliar fungicide on the shoot and expect that you're going to see a benefit down the root. They just don't get translocated down to the root. Seed treatments don't last long enough. If the disease isn't coming on until like mid-June, your seed treatment's active only for those first sort of 2 to 3 weeks at the seedling stage. We've looked at a lot of biological control options because the idea is that these should be able to grow with the roots. But so far, we haven't found anything that worked. We've looked at some soil amendments, liming, other things, and so far, we haven't found anything that worked. So Yvonne isn't here anymore, but she would get an A+ on her exam. The only thing that works is crop rotation. And when we talk about crop rotation, we talk about it for 2 ways. First, to prevent root rot, and then to avoid root rot once you have it. So prevention is you're interested in starting to grow peas, lentils, pulses. You know they're really good for your soil. You know root rot can be a problem. How do you prevent them from developing? And the only answer is rotation, rotation, rotation. So it's the frequency of susceptible pulse crops that are in your cropping history that's really going to determine whether you have a problem with root rots developing. Again, the problem is there's no magic number. You know, so we'd say like plant pulse crops 1 in every 4 years, and it might take you 5 cycles of that pulse crop. So you might have 20 years before you start to develop root rot. 1 in every 6 years, then if you think it's going to take 5 cycles, then it might take you 30 years. But there's no, unfortunately, there's no sort of magic number of knowing how many times it's going to take, but you want to start out by practicing good rotations at the beginning. No lentil durum rotations. We don't want to see that. And then the other big thing, like I talked about, is that soil moisture during your pulse crop. So you're in a bit of a tricky situation here because pulses kind of like water, but if you happen to grow peas, say in 2020, which was a really wet year, you're leaving more Aphanomyces behind in your soil. That means the next time you plant peas, you're going to have more Aphanomyces in your soil. So if you happen to have your peas in a dry year, you're better off. And then some of it's just kind of luck. And then you have root rot, so now what? Now the only option is kind of avoidance. And this is an extended break away from pea and lentil. And how long? I don't know. So it's a big space under there because it's another thing we just have not been able to figure out how long it takes for these oospores to die in the soil. And it all comes back to these pesky little oospores that can survive about 6 to 10 years. Seems to be kind of accepted, but sometimes we've seen longer, so it's really hard to determine how long. So what we're really moving towards, and I think I said in my intro, was I'm chasing this dream of being able to develop a soil test where we can quantify the soil-borne pathogens and compare them to the threshold levels that we know cause disease. So we've been working on this. A lot of soil testing labs do offer this service, but it is still a bit of an imprecise tool, and that's because those ooze spores that I showed you with those really, really thick cell walls makes them resistant to some of the DNA extraction techniques. And then each soil type, kind of clay loam, clay, sandy loam, they all have different thresholds, so you kind of have to know what kind of soil you're working with for the test to work. So what can you do now is really think about how your past pea or lentil crop did, and this will determine your rotation and interval and kind of your decisions for the next 4 to 8 years for that field. And then the caution is that 2023 was a really, really dry year, so you might not have think you had root rot, your peas might have done well, but those levels could have just been low and you didn't get disease. So I know, I just like bad news up here. So this is sort of what, just kind of to think about when your last pea and lentil crop, if it looked like this the last time you grew a pea crop, or a soybean crop for that matter too, you know, it was healthy, no yellowing spots, then you're pretty safe to plant a pulse in that field in 2024. If when you grew it, you started to notice these like yellowing spots showing up, usually in these water track areas, here, see the water track, there's a low spot in that field. If you notice those yellow spots, oh, sorry, jumped ahead. If you had a pea field that looked like that, really empty spots in your field, then don't plant peas or pulses in that field in 2024. If you just started to have those kind of yellow spots, those are the ones that are a little bit harder to know what to do with. So we do recommend soil testing those sites to try to get an idea of whether you have some of these soil-borne pathogens in your field or not. So where we're moving to with the research, and that's being supported by Manitoba Pulse soybean growers and all the other pulse associations, is, you know, we're trying to develop the soil test so you know what your pathogen levels are. Maybe work in some microbiome indicators if we can figure out, okay, one of those beneficial organisms that tell you that you have a healthy soil. And then also working towards patch management, where you see those little yellow spots and you know that that could be a problem in the future. And then superimposed on that would be, okay, well, how long do I have to wait? That I know what my ooze spore levels are. It's going to take 4 years, or no, it's going to take 6 years to reduce those ooze spore levels to the safe spot. And then stacked on top of that is the use of partially resistant cultivars, which are coming for pea, not quite there for lentil. If I talk to root rot, you're laughing, you have resistant soybean cultivars. Anyway, so I guess just my final inquest report is pulses are affected by many soil-borne pathogens. We're talking pea and lentil, Aphanomyces is the worst, followed by Fusarium, and then we have kind of these small bit players. Very, very difficult to link soil health properties with soil-borne pathogen presence. So as there's more research happening into soil health and soil health parameters, hopefully we'll start to tease that apart a little bit more. And then in terms of management, soil test, avoidance, and you can plant kind of soybean, faba beans as a safe pulse crop option. So finally, thank the funders and my collaborators. And then time for some questions.
Thanks, Syama. That was a whirlwind of information on the bad guys. We did a lot of talk about the good guys, and we're going to do some more, but there's bad guys down there. Anyone have any question about these bad guys that Syama can? Yes, sir, up at the top.
Oh, so for the soil test, you can test it. So fields that you're considering putting into a pulse crop for 2024, you can test those before you plant, and the inoculum will still be in the soil. So the soil test you can do as a pre-plant soil test. And most of the soil, I'm not 100% sure with Manitoba. You guys have 2020, is there 2020? BioVision Seed Labs here, right? They offer the soil test for Aphanomyces. Not as many offer a Fusarium soil test yet, but most of them do a Aphanomyces test.
Okay, I have yes, sir.
Oh, yeah. Yes. Okay. Yeah.
What triggers oospore
Yes,
germination?
that's a very good
Yeah.
question, and some of my colleagues at the AFC in London and AFC in Lacombe are studying that. So there is very specific triggers that come from the host that tell those oospores to wake up. So yeah, the idea is if we knew what those were, then we could try to take advantage of them. It's likely some kind of flavonoid. I think soybean is well known what triggers Phytophthora root rot, but we don't know for Aphanomyces. But yes, so there is a very specific host signal. Are actively researching that. There's this thing with fababine and how we don't see them be a host. There's some thought that they can trigger the oospores to germinate, but then Aphanomyces can't actually infect. So that's why we're thinking they could be they're a good preceding crop because they might actually trick the oospores into germinating. So a very good question and a very kind of active area of research to figure that out.
So I have a question. How do these little beggars get in the soil in the first place? So,
Yeah.
and where I'm coming at is from a biosecurity perspective. So when clubroot started to be noticed, the canola industry kind of put in place biosecurity to prevent its spread. But can we do the same thing with these?
Nope. That's why I'm always giving the bad news. No. So what's kind of thought of with Aphanomyces, first of all, is that, and I think Phytophthora would be the same. These are ancient marine organisms. So any area that was like covered in oceans at some point, the hypothesis is that you kind of have these really old signatures of Aphanomyces or Phytophthora in the soil. And so that's why we kind of see that Aphanomyces is already globally distributed. And so they're just sitting at really, really low levels in the soil, and then it's not until you start cultivating these pulse crops, well, particularly peas and lentils, that it kind of, boosts those levels. So it's not, yeah.
So how like older than me ancient?
Yes, yeah.
So like late Cretaceous?
Yes,
Okay.
I'm not a geologist, I don't know, but that's what I've heard other
So
geologists
our
say,
cropping
yeah.
systems are affected by something that happened 73
Yes.
million years ago.
Yes,
That's a little... Yes, sir, Vikram. Technical question.
Yeah,
Maybe
a
repeat it.
technical question about, and it's actually a really good question about being able to culture out these different root rot pathogens, because that's how we diagnose them in the first place. So Pythium, Phytophthora, and Aphanomyces are all part of the water mold group, and they are very difficult to separate out from each other in culture. What usually happens is that Pythium, like another reason why we didn't think we had Aphanomyces was part of this culturing issue. You would just get Pythium, 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 and you're like, oh, we don't have Aphanomyces, it's everywhere else but here. And that's because it's very difficult to separate them out. So we have to put some fungicides in our media that will kill Pythium but let Aphanomyces survive. And that's the same with Phytophthora. So you have to play around with these different fungicides in the media so that you kill one and then the other one can grow.
So one of the things that we've done, because this is a problem across the prairie region, we've collaborated with the other grower groups in Saskatchewan and Alberta and formed what we call a root rot initiative so that we can take a collaborative approach to figuring this out. And we've gone through a whole bunch of different exercises to determine just what kind of research should people like Syama and Youngmin Kim, who's here, and Yvonne on the agronomy side, what kind of things should they do? One of the things that we've discussed lately is because of the intense connection between Aphanomyces especially and water, how effective is the tool of drainage?
That's a really good question, and I don't have the answer to that one. Yeah, I think it could be effective, particularly in places like Manitoba, where you guys have more water all the time. In Alberta and kind of southern Saskatchewan, where we struggle more with dry conditions, I don't know if we'd got to look into drainage, but I think that could certainly be a tool in Manitoba. Yeah.
And a reminder, drainage is a regulated activity here in Manitoba, but there's more, we're noticing more soils getting tile drainage. And so kind of looking a bit like Ontario in some areas where they're doing a lot of tile drainage, and we'll follow that to see if there's any impact on soil, on soil pathogens. Any other final questions? Right there, yes.
Yeah. Yeah. Yeah, I think right now the biggest barrier is that our detection limit is the threshold limit. So the quantification tools aren't precise enough yet to be able to say like, oh, you have 55 ooze borers per gram of soil. And if you plant peas, your risk is like 60% of getting disease, or you have 210, right? So really all we can tell you right now is, yes, you have a Phantomyces, or no, you don't have a Phantomyces, without really getting into what the quantification part is. So that's sort of the barrier when I said it's an imprecise tool. It's because right now we can kind of be like yes or no, but not give you that quantification. And so we could say that yes, you have it, but you could have really low levels and you might be okay. But for now, usually if we say yes, you have it, it's like, well, just don't plant in that field because we can't tell you what the situation is going to be.
Great, thanks. Well, Syama, thanks a lot for traveling from Alberta. And again, Ag Days is making a contribution to the Manitoba Farmer Wellness Program in recognition of your presence here today. Valuable information.
Great,
Thank you.